base pairs on average is unique for each individual. Okay? So if you look at about 1,300 base pairs, you find one of these that's different between you and your person sitting next to you. But otherwise, we're a lot more similar than we are different, as you can see. Now, this is only one source of genetic variation in the genome. Okay, these single nucleotide polymorphisms. There's other sources of variation between individuals too. Sometimes there's major genetic rearrangements in chromosomes. Sometimes pieces are inverted. Sometimes they're copied. Some folks have extra copies of certain genes. Some folks have fewer copies. Some folks have, sometimes parts of chromosomes get removed through a deletion, through some sort of traumatic event, or just through replication errors. Some people have insertions. So this is just one version of genetic variation between humans, but there are others. Okay, so some of those other ones, like where you might have a piece of a chromosome deleted, that sometimes those can be quite significant in terms of human health or your phenotype. Okay, make sense? Okay. Okay, so we've talked about in these complications, we've talked about incomplete dominance, where the heterozygous has an sort of an intermediate phenotype. We saw for the flower color there. We looked at briefly codominance and multiple alleles. The blood types were the example. Here's another example of complexity of Mendel's basic laws. It's what's called epistasis. And the word is informative. Epi means like lid or top. And stasis, in this case, means to stand on. Okay? So, in epistasis, sorry, still recovering from my terrible cold. This is where the product of one gene and what's the product of a gene? What's that type of molecule that's the product of a gene? These, of course, are DNA. And what are the products of DNA? Starts with a P. Next letters are R, O, T, E, I, G. Remember DNA to RNA to protein. So a product of one gene, a protein of one gene can influence. You know what influence means, right? Yeah. The expression of a second gene. And so it's the interaction between gene products, and often you find these in metabolic pathways. The classic example of epistasis actually has to do with fur color in mammals. Actually, and also in skin color in humans as well, as we'll see in a bit. Not so much skin color, but that condition called albinism. That's due to epistasis, as I'll show you in a second here. Let's look at these beautiful dogs here. Three basic colors in these, some Labradors. We see black, chocolate, and yellow. In the metabolic, now the coloration of mammals has to do with how much melanin, that's the pigment, that's deposited in your skin, the iris of your eye, or your hair. Same pigment, the more you have, the darker you are. Okay? Now, melanin is derived from an amino acid called tyrosine through a number of biochemical reactions. And two critical genes that influence how dark you are in this metabolic pathway, there is something called usually referred to as the E gene and the B gene. Okay? Now there's the dominant and recessive allele here. Now, the B gene kind of really sets how dark you have the potential to be. Are you going to be like deep dark black, or kind of very light brown, or really pale face? So for a lab, if you have one dominant allele, you're going to be black. Okay? If you're homozygous recessive, you're going to be chocolate. Chocolate. Okay? Can I spell chocolate? It's my wife's favorite drug. She says it's a drug, and she can't live without it. I said, okay. She can live without me, but chocolate, between me and chocolate, I lose. I know that. I accepted that when I married her. It was a wedding agreement. Till death do us part, I ran out of chocolate. Okay. All right. So this is really the amount of pigment deposited because of this gene here. This E gene determines whether the pigment is deposited or not into the fur. Okay, so this is how much you make, how much pigment synthesized, how much melanin is synthesized from tyrosine. This determines whether it's deposited or not in the fur. If you have a dominant allele, in other words, if you're homozygous dominant or heterozygous, and you have a big B gene, you're going to be chopped black if you're a Labrador. If you're homozygous, if you're homozygous dominant or heterozygous for E, and you're homozygous recessive for B, you're going to be chocolate. But if you're homozygous recessive for this E gene, you deposit almost no melanin, and you come out, you're yeller. Okay? And so the E gene is epistatic to the B gene. The gene product of this will determine whether or not the B gene, depending on what your should be, is even deposited or not. And the clue that there's an epistasis happening here is there's a distorted F2 phenotype ratio of a dihybrid cross. Now, I didn't review dihybrid crosses because I don't want to spend too much time and want to get this chapter done quickly. But if you remember Mendel's dihybrid crosses, he found that the F2 phenotype ratio was 9 to 3 to 3 to 1. So when he crossed yellow round seeds with green wrinkled seeds, he got all yellow round at F1, and at F2, he got these 4 phenotypes, I think, yellow round, green wrinkled, and yellow wrinkled, and green round. These are the kind of their components here. 9 through 3. Remember that? If not, go back and look. The evidence that there's epistasis, where one gene product is affecting another, is when you have a distorted F2 phenotype ratio. You end up with something like, instead of 9.331, you have something like 9.34. That's telling you one gene product is affecting another. Where the E gene, you only express the B gene or deposit the melanin in your fur if you've got a functioning E gene. If you're homozygous recessive, you're kind of, if you're a human, you're an albino. If you're a Labrador retriever, which none of you are, I just noticed, you're yeller. Okay? So this is from the textbook. So here's our 2 true breeding homozygous parents, black and yellow. Notice if there was a dominant E here, the dog would be chocolate, right? And here the functioning E, you've got the black pigment, a lot of melanin deposited in the fur. Okay? You use an F1 hybrid. Well, they're still black because of complete dominance of the B gene, and you've got the functioning E gene here, so the melanin is deposited in the fur. Now when you allow the F1 puppies to reproduce, here are the gametes from each F1 dog, okay? All combinations of alleles, right? For both pairs, we're going to have a 4x4 Punnett square, just like a Mendel dihybrid cross. You with me so far? I haven't thought about that for a week or so. There's from the other parent. And we see that as long as there's functioning a dominant E allele, we can have, depending on the B gene, we can have, if you've got a dominant B, we can have black dogs, okay? And all these have a dominant B and a dominant E. 1, 2, 3, 4, 5, 6, 7, 8, 9, okay? But notice, and we got 3 chocolates here, because they're homozygous recessive for the B allele. Minimal melanin production, but at least it's deposited because of the dominant E allele. But then, instead of only having one homozygous recessive like the recessive parent, like at a 9 to 3 to 3 1 ratio, we've got 4 of these, because all 4 of these have the homozygous recessive condition for the gene that deposits the melanin in the fur, and they all come out yellow. So it's 9 to 3 to 4. So the E gene is epistatic on the B gene. All right. This dog with a functioning E gene, it would be black. Be black. Should be black. Should be chocolate. But they're all turn out yellow because this gene product was not present to allow this allele to be expressed. These alleles to be expressed. Epistasis. One gene influencing the expression of a second gene. That make sense? So it's a complication, another complication of Mendel's laws. Am I going too fast? I feel like I gotta go faster. When I get behind that, I really like the truck. I'm gonna erase this over here, is that okay? People are laughing. I zipper up? Yeah, okay. All right, let's look at the next complication of Mendel's basic laws of inheritance. Actually, I'm sorry, let's look at the example of epistasis in humans. Again, there's an enzyme that one of the enzymes that converts tyrosine to melanin is a tyrosinase. Okay, and there's a variant there where there's a mutated version of that gene that gives you a non-functioning enzyme, and in that case, it doesn't matter how much melanin you would have produced based on your sort of family background, you become an albino through epistasis. You can see there's no melanin in the irises to be pink, no tone at all, white hair. Okay, so mine used to be blonde. I just showed this guy because this is one of the most famous Texas blues guitar players of the 1960s. Both him and his brother were albinos. That's how these guys play a bunch, because they were just tremendous blues guitar players. And like, what's the blues? You don't even know. But interesting, their last name was Winter. I thought that was a cool name to have. That was their real name, was Winter, having this white hair. That's the great